In this video, we will discuss about what are the approaches, glycomics approaches that are used to detect congenital order of glycosylation. So this is a family a family of diseases in which the glycosylation post-translation modification got affected, and because of this, the patients will have the different glycan profiles, and these different glycan profiles can be detected by two different technologies. So before going to the technologies, let us quickly revise how this glycosylation process occurs within the mammalian cells. So this glycosylation process begins in the endoplasmic reticulum with this glycan precursor, that is bind to the dolichol precursor, is present within the endoplasmic reticulum, and it will bind to the translating protein uh, at the aspargine site, and after that it will get cleaved by these many glucosidase 1 and glucosidase 2, and simultaneously the protein folding will occurs, and this glucosidase 1 and 2 will trim off this glucose structure one by one, and if the protein folding is successful, then they will move to the Golgi for the post-translation modification. And some of the post-translation modifications are shown here, such as some glycans will form these high mannose glycans, whereas some acquires this hybrid glycan with this glycneck along with this mannose, and some of the glycans will acquire this complex structure that includes all the glycneck, that includes glycneck, mannose, glycneck, galactose, and also the sialic acid. So this is just the normal glycan profile that is shown here. In the case of congenital order of glycosylation, any of the process get disrupted, that might lead to a different glycosylation profile, and uh, this needs to be identified by some kind of experimental techniques, and there are two different experimental techniques that are available to identify the change in glycan profiles. One of them is to use this PNJs. What this PNJs does, it will cleaves off the glycan at this aspargine site, so that we can get the information about the information about the intact we can get the information about the glycan structures only. So that can be seen here in the total plasma glycoprofiling. We will get the information about the glycans without the backbone protein chain. So, and there is another approach that can be used is to use this immunopurification. So in this case, what can be done is we can uh, selectively identify or separate only those glycan structures separate only those glycan structures based which are of our interest, based on these antibodies that can target only the glycan structures of our interest. So in this case, the glycans will not only get separated, but it will also take its it will also uh, take the protein backbone chain with its, that can be seen here in this bottom intact transfer in glycoprofiling figure. In this case, the antibodies detects these glycan structures, and then this glycan will also take the protein backbone with its cells. So in this case, when the sequencing will occurs in the mass spectrometry, we will get the glycan structure. At the same time, we will also get the sequencing of the backbone. So this information will provide us the information. This profiling, this type of glycoprofiling will not only provide us the information of the glycans of our interest, but it will also provide the information at which side of the this peptide or the protein this this kind of glycosylation are mainly found. However, that information is lacks in the case of this total plasma glycoprofiling. The other advantage of this, however, the advantage of this total plasma glycoprofiling is that you will get the glycans, total glycans that are present within the plasma, and not just few glycans of our interest. So both the techniques has their own advantages and disadvantages, and it depends on the final results that the uh, researchers are researchers wants, whether they already know about, whether they are expecting some kind of glycan uh, some kind of glycans that are highly expressed, then they can use this intact transfer glycoprofiling. And if they want to know the whole glycomics profiling of the plasma samples, then they can go for this total glycoprofiling. So in this case, so in this way, the different glycomics profiling can be used to identify this congenital order of glycosylation. So if you need to understand more about these techniques, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.